involving manipulation of light, heat and forces with metallic nanoparticles. And I hope with the background I gave you in the first lecture you will be able to get the most of them. So the first one is about optical antennas and how they can direct the emission of a single molecule. So this is related to what I told you about directionality and photonic mode density and how the photon emission is influenced by the presence of metallic nanoparticles. Okay, and this was done at ICFO with this colleagues, Steve Tavigno, Franz Segering, and Nick Van Holtz. So what is an antenna? What is an optical antenna? Okay, you know antennas from common life, they have diverse designs, but in all cases an antenna is a mediator between far field radiation, between propagating radiation and local fields. That's what an antenna is. And these local fields, in optics we call them near fields. Okay? So in a microcircuit, for example, if we want this circuit to interact with radiation better, what we do is we put an antenna into it. The simplest antenna is this half lambda antenna, and if radiation arrives to this antenna, will generate currents, and will drive the circuit in some way. So that's how the interaction between this guy and far field radiation is enhanced or controlled. So now we want to do the same, but for a molecule. So you know molecules also interact with radiation. We know the electronic transitions can be done by light absorption, and we would like to control that or to enhance that. So we cannot wire an antenna to a molecule, but what we can do is use the metallic nanoparticles, which are resonant at optical frequencies, the same frequencies that molecules absorb light. They also concentrate fields, so these local fields are in particular positions of a nanoparticle. If we have a rod, then a dipolar resonance, then that's very simple. The field will be concentrated at the tips. So what we have to do is to place a molecule in those regions, and then we will have a coupled system, like in this circuit. We will have a molecule that is coupled to this optical antenna. Nice, but experimentally it's not so easy to do that. So the way we did it was by means of a scanning probe microscope. So we used these tips. These tips are made of glass optical fiber that we pulled until it breaks in a very, very sharp tip. Then we evaporate aluminum all around. And then by focused ion beam, we sculpture the apex of this tip in order to leave a little aperture of glass, so that, I don't know if you can see, that dark circle at the apex is glass. And we also leave a protrusion of aluminum. Here you can see it from the side. And that is our antenna, which we have to actually tune to 1/4 of lambda, not half lambda, because the other half would be a reflection in this bulk metal. Okay? So the experiment then, once we have this, which is not so easy to fabricate, the experiment is relatively conceptually very simple. What we do is we couple light into this optical fiber. Some light will leak out of this aperture, and if it has the right color, it will excite a resonance on that metallic nanoparticle. And then we will have some fluorescent molecules that we will scan over different positions near that antenna and collect the fluorescence light of that molecule. Okay? So if we do that and we see what happens when the molecule is excited by the near field of this antenna, we see the fields are highly confined. You see it's 25 nanometers, so the antenna is working at the right. But to make it work right, of course, it was not so easy because we had to tune the length. You saw before in the last talk that the resonance, the frequency of the resonance depends on the shape, on the size, on the material. So okay, we have aluminum, we have this diameter of this rod, and then we have to tune the length to the right color of the excitation light. And then or the emission line of the molecule, depends on what we want to do. So we have to test different lengths, and we saw the resonance with quite some scatter in the data, but still the resonance is visible, and so we can pick the right antenna for our molecules. Okay, now to what we want to test. I told you that molecules, when they emit light or absorb light, they act as a dipole, right? The electron that goes up and down in electronic states is like we can think of it like a classical dipole. So a molecule has this dipole fixed on its molecular structure and will emit light like a dipole. So if we look where this molecule will emit photons, we will see that it will emit photons, a lot of photons in this direction, and nothing in this direction. Now we take a metallic particle, a rod, like the one we are using here. If we take a look at how this guy scatters light, when it's resonant along its main axis, we will see it scatters light also like a dipole, but with the opposite direction, so 90 degrees rotated with respect to this molecule. So now what happens if we put that molecule here, right? And we calculate this. We can calculate this is a simple calculation, and we see the result is this. So what's going on? If we put this molecule in the right position with respect to a metallic nanoparticle, we can rotate almost by 90 degrees the angular pattern of a molecule. So the same molecule free will emit in this direction, the molecule coupled to the antenna will emit in this direction. How can we test this experimentally now? So, okay, we can perform more detailed calculations now, including the sample, substrate, the interface, and some distances and the right collection angles, etc. So more realistic calculation. And we see how if we put the molecule in the right position, it can couple to the antenna. So if you take two snapshots with 90 degrees of phase difference, you see the system is really resonant and they are working together. And now, if we take a look at how the light is emitted by this molecule, I told you it acts like a dipole, right? And when you said, yeah, nothing here looks like a dipole. That's true, but because we have an interface now. So these molecules are embedded in a polymer, right? So we have an interface of polymer and air, and there light can be reflected. So what happens in the end is that if we look how a molecule oriented like that, parallel to the interface, emits light, it looks like this. So to the upper semi-space, it emits like a lobe, like a dipole, but to the lower hemispace, it emits in two lobes that are more or less showing you the total internal reflection angle of this interface. Okay? So this would be a free molecule in our sample. And now we make this distance shorter to the antenna. And we take a look what happens. We see that the angular pattern of emission is changing until we reach this situation where the light is emitted with radial symmetry along the antenna. So what we see is that here the light emission was dominated by the molecular dipole. Here light emission is controlled by the antenna emission pattern. If we look now how this looks in a microscope, now this would be like the back focal plane of a microscope. Basically, we are projecting these two lobes on our microscope and we see them, right? And we see how we get closer to the antenna and couple the molecule to the antenna, then the light comes in all directions. That's simply because now the dipole that is emitting is the antenna, which has a total rotational symmetry with respect to the interface. Okay, so this would be one way to measure that experimentally. We didn't have that possibility, but there's another way by means of polarization. If we look at how light is polarized here, most of the light is polarized along the dipole direction, as one would expect, right? Molecule is emitted like that, okay, there is total internal reflection, it deviates light to some angles, but the polarization remains. Now, as we approach the molecule to the antenna, we see the polarization state changes, and now, in this case, we have radially polarized light, because the light is going through the antenna, which is, again, symmetric in all this axis. Okay, and this is a situation that is easy to distinguish experimentally, this one from this one. What we have to do is now repeat the experiment, but now place here a polarizing beam splitter and two detectors. And what one would expect is that if light comes polarized along one direction, all the light goes to one detector, and if the light comes radially polarized, it should go equally or more or less equally to both detectors. So we did that, and we obtained this kind of images. So let's interpret this now. These are the fluorescence signals of individual molecules randomly placed on this sample. Okay, the molecules are immobilized and randomly distributed with random orientations. I circled here many signals of single molecules. And what you see is usually, and the color here, the polarization is color coded. So in red we have the signals from one detector, and in green the signals from the other detector. And yellow means an almost perfect balance between red and green. What we see is that for each molecule we have always, or almost always, a red big signal and a small yellow signal. The red big signal, big is relative because actually that is 100 nanometers, comes from the excitation of the molecule by the aperture. Okay? Some light leaks through the aperture with the right polarization, and it will excite molecules that are oriented along that polarization. In that situation, molecules are not coupled to the antenna and will emit with the same polarization, they look red. Now, the molecules can also be excited by the field at the tip of the antenna. In that case, molecules are coupled to the antenna and will emit light through the antenna, and what we see that those signals, that's those very small signals, which are like 25 nanometers in width, come always with equal polarization in both detectors, showing that the light goes through the antenna now. What is nice about the experiment is that we have, for the same molecule, with a given orientation, we have a signal far from the antenna, excited by the aperture, and coupled to the antenna, for the same molecule. And we can see how the molecule, when it was 100 nanometers away, was emitting red, if you want, polarized in one direction. When we move it 100 nanometers closer to the antenna, now the light comes polarized in all directions, because it's coupled to the antenna. It's the same molecule, so it's a self-reference measurement. And what's also nice is that we see different patterns. So if you look, for example, here, we have a small signature on this side, the red signal, and then separated the small signal again. We also have some signals that are we only see the yellow ones, and in some cases, for example, see here it was separated from the red, here it's like inside the red. So we have some different patterns, and the reason for that is the different molecular orientations, and can be understood very simply in this geometry. Take a look at this. So we have our nanoparticles like a rod with a dipolar resonance. So this would be a scheme of the field lines. Now if we have a molecule now parallel to the antenna, and we scan it, it will only interact when it's exactly underneath the antenna. And it won't interact with the field of the aperture, which is polarized in the other direction. So these molecules only give you the small signal. That will be this one, that one, that one. Those are molecules that are oriented like that. Now let's think of a molecule that is like this. This molecule will give you a small, let's say, will give you a yellow signal coupled to the antenna here, and also will be excited by the aperture. So this would be, for example, this case, or this case, right? Now if you have a molecule with intermediate orientations, it will couple only in one side of the antenna. So a molecule like this will couple to the antenna only here, and not here because it's perpendicular to the field lines. So these guys will show up with this, if the aperture is here, as we are imagining, these guys will show you the red spot away from the red one. That would be something like, in this case, okay, and the opposite will happen if the molecules are like that. Okay. So analyzing those patterns, one can even calculate or determine the molecular orientation with some degree. So here you have more publications on this subject, and this is the team that we worked with. It was Franz, Nick, and Tim. Okay, so here's some papers, and also in this website I try to keep updated with publications that we do. Any questions about the antenna experiments? Because now we move to a different area. All right. So now I will tell you about using the localized surface plasmon resonances to detect biomolecular interactions. So, okay, this is about an emission assay for cardiac troponin T. This is a protein that is quite important in diagnosis because it's a signature of damage in the heart muscles. So every time the doctors find somebody lying on the floor, they want to check if he had a heart attack, so they make a test of troponin T or some other troponin to check the heart condition. So, and what's the idea here? It's very simple. So we will have a gold nanoparticle with functionalized with an antibody, and another antibody functionalized with the fluorophore. Both of these antibodies are different, but bind to the same protein, just on different locations. So that if this protein, the troponin T protein, our target, is present, this molecular sandwich can form. And as we saw before, if we put the fluorophore very close to a metallic nanoparticle, the fluorescence can be quenched. And so we can detect this interaction by a decrease in the fluorescence signal. That's the idea. The advantages is that okay, we don't need to label the analyte because we form this sandwich, and it's an homogeneous assay, so there are no separation steps involved. And another advantage is that this quenching occurs up to 15 nanometers or 20 nanometers, which is long in comparison to Fred, for example. And since proteins and antibodies are rather large molecules, it's convenient to have a mechanism that acts over longer distances. Okay. Okay, this is another. This is probably related to what I told you about the photonic mode density. I told you that metallic nanoparticles can quench fluorescence by direct energy transfer that takes place very close to a distance of 10 or 15 nanometers. Over longer distances, the emission of a molecule can be enhanced or suppressed by changes in the photonic mode density. What's the photonic mode density? It's the density of photonic states. So where will give you more, a higher photonic mode density will give you higher chances for photon emission. That you can calculate simply by electric fields at the emission wavelength. Another way to see that in simple geometries is like this. For example, if we have a sphere now and a molecular fluorophore with a certain dipole. So this molecule has a dipole like this and will induce in a sphere a dipole in this direction. So these two guys will work together on the same direction, and then the emission will be enhanced. But now if we consider this other possibility, now the molecule is 90 degrees rotated to the nanoparticle, the induced dipole in the metal has this direction, and now you have these two dipoles working against each other. So in this case, the fluorescence will be suppressed. Okay? It happens, as it was mentioned before, in our case, we consider all this, and since we have random orientations, basically what we have is like 2/3 of this and 1/3 of this guy, and the effect is like a very slight increase in a decrease in the fluorescence. So, but it's not the main contribution. The main contribution here is the direct energy transfer. And okay, for that, as you can imagine, we need dyes that are quite good so that we can destroy the quantum efficiency. If the dye is very bad, there's little we can do. So we tested different dyes to prove that. And then we made to make it short, we made this thing work, one has to be careful because if to adjust the concentrations, because if you add too much of the protein, then you destroy the sandwiches because the same protein starts to compete for particles or fluorophores. But the advantage is that if you adjust the concentration of particle and fluorophore, you can produce a sensor that is less sensitive but has a large dynamic range, or a sensor that is more sensitive with a less dynamic range, depending on the application that you want. And this is the real measurements, and you see it works, and we reach limits of detection that are quite nice for the people that work in diagnosis. Okay. Another example of biosensing, and this one is even simpler because it's based, you saw that these resonances depend on the environment of the nanoparticle. If we measure the resonance of a nanoparticle in water, it picks at one wavelength. If we measure it in methanol, it picks at another wavelength. So if you change the dielectric environment of a nanoparticle, the resonance shifts. So now we're going to use that effect to detect biomolecular interactions. And we did it with just to test some new particles with these guys that we call them nanostars, which are, it's like a sphere with a lot of protrusions in different directions. And these ones, with this complex geometry, have a lot of resonances. Here you can see the resonance or the resonances of one of these as a function of the polarization. So if you change the polarization of light, you start making resonance in this direction or there, and they have different sizes, and the resonances are different. So here you see the peaks, the amplitude of the peaks as a function of the polarization. And here you see the dependence on the environment. So here we change the refractive index of the environment, and we see the resonances shift. So we can use that now. If we functionalize those particles with the right biomolecules, we can use that effect to detect the molecular interaction. So we did like, we cover the surface of these nanostars with a protein that has a moiety called biotin, and that biotin interacts very strongly with another protein that's called streptavidin. So we want to detect this interaction by looking at the resonance shift. And so here you see one example. This is done now on a single nanoparticle. So we look at one nanostar, we measure the spectrum, we add biotin, and we see how the resonances of that individual nanostar shifts. That's what you see here. And okay, we can follow the shift of the different peaks, and we see, of course, that the longer wavelength peaks shift more, because it's just like a grating effect. And then we can titrate the sensor. This is a single nanoparticle sensor, and we see that we can detect very low concentrations of this product. Okay.
Question.
Yes.
Are those nanostars made of gold? Or
They are made of gold, yes. But to make them, you need to add, you know, these nanoparticles are all made by nucleation and growth methods. In this case, it turns out that you need to add a small amount of silver ions in the solution, and that happens. They start to branch. But the amount of silver is negligible. It's like a catalytic effect. So they are mainly made of gold. Any other questions about the previous experiments? Okay. So now we go to heat. And as I promised, I will show you now an example about how to use the nanoparticles as local nanoscopic sources of heat that we can switch on and off very simply with a laser, and try to obtain local thermodynamic information with that. So why do we want to do that? Okay. Nowadays there are a number of microscopy techniques that can give you impressive resolution. Now you can know the structure and pathways of nanostructures and nanoscopic systems with high resolution. But still, a functional system, if you want to understand it in detail, you need also some energetic information. You need to know those processes, how much energy they take, and from where, etc. Traditional thermodynamic experiments are made in bulk samples, it's like differential calorimetry, etc. So you heat up a whole system and take a look at how the system absorbs heat or releases heat. So we are interested now in exploring the idea of doing thermodynamic experiments locally at the nanometer scale. So for that we have the basic tool, which is a source of heat, our nanoparticles, and the rest you have to figure it out in each case, you will see. So in this case we started working on phospholipid membranes. So here this cartoon shows you the idea of the experiment. We will have a phospholipid membrane and we will heat it locally with nanoparticles in order to produce a phase transition only in a small region of the membrane. So why do we want to do that? Well, phospholipid membranes are the membranes of living cells, of all living cells. And it's a very complex system that contains a lot of proteins and receptors and biomolecules, and a big number of vital processes take place there, like photosynthesis, nutrient recognition and uptake, ion transport and signaling. So it's a very complicated system. If you would like to do thermodynamic experiments on that, like increasing the temperature and taking a look what happens, you will have such a number of processes going in different directions that it's impossible to get some information. So that's why we thought, okay, let's start in this direction. This is a nice system to do local heating and see what happens. But of course we started first with the model system, which is an empty membrane. These model systems are called giant unilamellar vesicles, and it's like a big bubble of a phospholipid membrane. So here you see a dark field image of one of these GUVs decorated with some gold nanoparticles on the surface. This membrane is only 5 nanometers across. Here it looks wide, it's just because of the resolution of the microscope. But it's a very thin membrane, and we put nanoparticles that are quite large, are 80 nanometers here. Okay, and we will detect the things with a dark field microscope, like I explained to you before, and we add to that microscope now an additional laser that we use to heat up the nanoparticles. Okay, so the setup is very simple. Here we have an example. This is one of these GUVs labeled with the fluorophore and decorated with gold nanoparticles. And we wanted to test if we can really heat up this thing. So the experiment starts. We try to catch one nanoparticle with our laser, and once we do it, like now, after a couple of seconds, the vesicle is destroyed completely, and we are left just with a blob of phospholipids and nanoparticles. So this is what people are using to do controlled release, to kill cells, etc. That's how you destroy things, heating up metallic nanoparticles.
What is the size of it?
Huh?
The size of that?
The size of this, it's like 20 microns. The GUVs are typically 10 microns that we use here. Okay, so we can destroy things if we use enough power. But we want to do things mildly. And this is what we want to investigate. These phospholipid membranes have a temperature sensitive phase transition. They can go from a gel phase, where all the phospholipids are tightly ordered because of a strong interaction between these alkyl chains. So from this gel phase, they can go to this fluid phase, where the interaction between the alkyl chains breaks up and the phospholipids can move, and the self-diffusion of them is 1,000 times higher in this case. So upon melting, this membrane becomes more flexible, and the self-diffusion is 1,000 times faster. And of course, the transition temperature depends on the kind of lipid. In particular, depends on the alkyl chain, which is the one that holds the guys together. So here you see they all have the same head with different chains, and the temperature can go from -20 to 42 degrees. Here another example. So now we produce these phospholipids, these GUVs. We produce them in the gel phase. So the one you saw before was round and nice because it was in the fluid phase. And of course it goes to the spherical shape, which is the most energetically convenient. If you produce them in the gel phase, the GUVs have these weird shapes, faceted and not so nice. And now we are going to illuminate only this area. And after a short time, we see that this guy can relax to the spherical shape, while the others remain unchanged. So it is possible to heat up mildly without destroying them. Alright, but still here there's nothing nanoscopic. Melted the whole vesicle. So now we want to do it on a single particle. So I don't know if you can, probably no, you cannot see it very well, but trust me that here in the black area there is one of these GUVs in the gel phase. And on its surface it has 3 gold nanoparticles, the ones that you can see here. Okay? And now we are going to illuminate only this guy. The other 2 serve us as a reference, for in case something happens. And now the experiment looks like this. So nothing happens until we switch on the laser now. And then this guy, the nanoparticle that is illuminated, starts to move. The other 2 remain fixed. Here you can see some of the vesicle. We do it again and again. This guy moves. We switch off the laser, it stops. Okay, we switch on the laser again, it starts again. And the other 2 remain always fixed. So what's happening is that we have the gold nanoparticles fixed on the gel phase membrane because they interact strongly with the phospholipids. So now we have our heating laser spot, and if a nanoparticle is inside of it, it absorbs light, it heats up, and it can melt a small region around it. When it melts, now it can move together with the lipids, and as it moves, it melts a new region, and so on. So basically the nanoparticle could move as much as it wants inside the heating laser spot, while the other 2 remain fixed. So we always use vesicles with 2 nanoparticles at least that serve as a reference, and then we can calculate these kind of trajectories. And from the trajectories, we can get the mean square displacement and then the diffusion coefficient. So you see, these are measurements on the same particle, on the same membrane as well, at different laser intensities. And we see that we increase the laser intensity, the nanoparticle moves faster, as expected. And we also see that at one point we see a subdiffusive behavior because the nanoparticle is reaching, in this case, it moved so fast that it could go to distances, it basically reached the border of the laser spot. So you have to think this is a Gaussian laser spot, so in the center, the nanoparticle is very hot. If it moves to the periphery, the intensity is lower and becomes colder, so it moves slower. Of course, we can do this many times and with many membranes and with many particles. And what you see here is the average diffusion coefficient measured in this way for different membranes. This one is made of DOPC, which has a melting temperature below ambient temperature, below 0 actually. And we see that if we increase the laser intensity, the nanoparticles become faster with some slope because of the viscosity changes and etc. In this membrane, which has a melting temperature of 42, we see a phase transition. So at high laser powers, we see the same regime like in the fluid membrane. So basically the nanoparticle feels free. In that case, the fluid area is big enough. But if we reduce the laser power, then we see the phase transition, and here this scatter is just our error in the position determination. This scale is logarithmic, so this is not that big actually. And if we change the lipid with a higher temperature, we see a different slope and a different melting temperature, as expected. The nice thing about this experiment is actually this slope. That is giving you unique information that you cannot get otherwise, because you have to imagine if the phase transition would be infinitely fast, the nanoparticle has its own lake around so it can move, but if it moves fast, it reaches the border of the fluid phase and has to wait for the phase transition to take place to open up a new lake around, right? If the phase transition is infinitely fast, the nanoparticle will never feel that. As soon as it reaches the border, there is a new melted region there, and it can move freely. So that will give you a behavior like that. So that slope, actually, you can calculate from that directly the speed of the phase transition front. So that's the speed of the fluid front once you give a temperature shock. And then you can determine from these kinds of experiments on a nanometer scale. Okay, this is another application. As you saw, the nanoparticle cannot escape the illuminated area, so it's a Corralled diffusion. It's a random walk, but in a limited area. And now if we move that laser spot, we can try to move the nanoparticles where we want, which could be handy if we would like to, in the future, to heat up different positions of a cell. And here you see we brought this guy like that and like that. It's not like a straight line because it's a random walk, just confined, but you can do it. Of course, if you're unlucky, you move the laser in one direction, then a nanoparticle decided to go backwards and you lose it, but you can always pick it up again and take it to the position you want. Here you see another example where we move this guy from here to here. These two are our references. So it started here, it finished there. And we also saw the positions on the laser spot. Okay. I think that's the end of this story. And now the future is, of course, try to move this scheme to a real cell and try to get some thermal information about real membranes or some trigger thermally the function of some proteins that are temperature sensitive, warm up, energetic obstacles for protein diffusion. We could give some protein a thermal kick to jump obstacles and measure like that the energy of some barriers in a real cell. So in order to try to get to cells, we did you need to do some more chemistry. This has nothing to do with the optical properties, but just to tell you that one can do many things on the surface of nanoparticles to really bring them to the surface of a cell. It's not just a crazy idea. In this case, what we did several steps in order to get a small vesicle of phospholipids where we had bound to it only one nanoparticle. So in the end, we had a big dispersion of vesicles where they had either none or one gold nanoparticle. And then if we incubate those vesicles with the cells, by membrane fusion, we can deliver that nanoparticle to the cell membrane. So that worked, and we could follow it in the real cell. All right. Any question about this temperatures? Yes.
If you look at the statistics of the random walk of a nanoparticle,
yes.
Does it follow what you would expect from normal Brownian motion?
In that case, no, it's a two-dimensional random walk because it's confined to the surface of the vesicle. And okay, the vesicle is spherical, but it's rather large. And our laser spot is 1 micron, and this 10 micron thing, so it's almost like a plane. So we don't see any deviations. What we saw was that subdiffusive behavior if you hit strong enough. So the nanoparticle basically gets to the edge of the illuminated area.
I was curious if in this case you have some optical forces acting on the particle.
Yes, you have.
In addition to the just viscous drag. So,
yeah.
So how do you separate the two? Is one of them more strong than the other?
We found that in these experiments, here the optical forces were negligible because the laser was quite large. So this is more or less the laser spot. It's large, it's 5 microns. In these experiments, we focused the laser more tightly in order to guide it where we want, and And in that case, the optical forces were not negligible. And in fact, what we saw is that this is what you see here at the position of the nanoparticle in the spot. And we see that we are kicking it actually. It's like it's pushing it away from the high intensity region. So that, yeah. Of course, this is a good question because all the effects that I told you in the first lecture and now that we separate into light, heat, and forces, in fact, they all take place at the same time, always. So sometimes one dominates, and that's what we want to focus on. But if you want to heat up, there will be optical forces, and there will be some electric fields that may do other things. So they always take place all together. Okay, and I think that's the last one. And this one is involving forces. And in this case, we are applying forces to gold nanoparticles, not for trapping, which is the usual case. Here we want to push them. Basically, we are using the laser just to hammer nanoparticles against the surface and print them on specific locations. So what's the idea? So with all these nanoparticles, most of them are made in suspension, so they are colloidal nanoparticles. That means they are stabilized in solution by electrical charges. So you have all nanoparticles with some charge at the surface, and by electrostatic repulsion they remain stable in suspension, right? Now, if you place a substrate with the same charge as the nanoparticles, then you will have a similar interaction, a similar colloidal interaction. The nanoparticles will not go to the substrate, they keep distance due to an electrostatic barrier. So this is how the potential looks like, right? So you have an electrostatic barrier here in red, and at very short distances you have a van der Waals interaction. So the resulting potential, it looks like this. You have an electrostatic barrier, and if you manage to go over it, look, you get in close contact and the things get fixed. Normally you want to avoid that so that the nanoparticles don't agglomerate to each other and you have a nice suspension. So here, what we are going to do is to apply an optical force strong enough to overcome this barrier and then fix the nanoparticles on a specific position. So the way we do it is, you see the setup is very similar to the previous one, but now instead of using a laser to heat up, we use a laser to push the nanoparticles against the surface. And let's see how it works. So what you see here is a dark field micrograph of gold nanoparticles, 80 nanometers in size, that are swimming freely in suspension. And we will start. Oops. I showed you the end of the movie before. And we'll start printing them one by one by the laser spot. You cannot see the laser here because it's filtered, but not to destroy our eyes. But so basically, wherever there is a nanoparticle, that's where the laser spot is. And we move the sample, ting, 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 and we can print gold nanoparticles one by one on specific locations. It looks ugly because it was done by hand by a student because we didn't have a nano positioner, but we hope to have one soon. So this is not the real precision of the method, it's just how patient the student was. But still, the system works. It's very easy, simple, and fast. So this is real time. So, all right, let's finish it. If you do it with more care, you can get it nicer, but still it's done by hand. It doesn't. Yes.
So you just hold the laser in one spot and then wait for a particle to go by, and then
That's correct.
it goes by.
That's correct. So the particles are swimming by Brownian motion in the solution. When by chance one gets into the laser spot, it's trapped radially by the optical forces. So that pulls the nanoparticle to the center, and it's pushed by the scattering force towards the surface. And that gives you your localization accuracy. But it's by chance, so that's why you need to adjust in this way. You need to adjust the laser intensity and the nanoparticle concentration to have a printing rate more or less reasonable. Otherwise, it's too slow. Okay, so this is basically what I just told you. You have, if you calculate these forces, this is done with a Rayleigh approximation, but using instead of a plane wave, a Gaussian beam, and you see the radial forces will push the nanoparticle to the center, the scattering forces, and in the z direction, the absorption and scattering dominates and pushes the nanoparticle against the surface. So the printing accuracy is determined in this case by the beam size and intensity, and by the Brownian motion. So that can be, of course, controlled. You can increase the viscosity of the solution. You can actually, since you have to overcome this barrier, if you use a very low laser intensity, you will print nanoparticles only at the very tip of your Gaussian beam, right? Because that's where the intensity will be strong enough to push them. In that case, you could increase the accuracy principle as much as you want, but of course it takes forever to print a particle because you have to wait until one swims by chance to that spot, right? That's why in the end you end up with a compromise. But there are many ways to change that. This is just starting. Now we can put some micropipettes to inject nanoparticles into the focus instead of waiting for them to swim and play with the viscosity of the medium. There are many easy parameters that you can play with. So here we try to see how accurate the method is. So we didn't have a nano positioner, but we had the stepper motor in one direction. So here you should not pay attention to the interparticle distance because that's whatever. Just need to pay attention to the target line. So you see some of them are closer and farther away from each other, but they aim pretty well to the target line. Here you see a zoom. So we computed the deviation in this direction from the target line as a measure for the accuracy. And we see that if and also to test if this mechanism was correct, because we changed the substrate by adding more and more layers of polyelectrolyte to change the electrostatic boundary. So we see that as we increase the number of layers, we need more laser power, so that means the electrostatic barrier is larger, and as we do that, also the precision becomes worse. But in the best cases, we can get an accuracy of 50 nanometers, which is quite good for such a simple method, considering the diameter of the nanoparticles is 80 nanometers. And I think this can be improved at least 5 to 10 times very easily. Okay. So this was done as a proof of concept with gold spheres, but of course it can be done with any kind of nanoparticle because the mechanism is not related to the plasmons here. It's just a colloidal interaction and optical forces. Any nanoparticle that can absorb or scatter light enough, we can do that. So in the future, and also, especially metallic nanoparticles, we can play with the orientation. One can orient, for example, metallic rods along the polarization axis and try to bring them in some certain orientation. So now we are trying to use this technique to build circuit of nanoparticles of different kinds and maybe test those nano antennas I showed you before, but built in this way and place a quantum dot in the right position, etc. I think there are many opportunities. Okay, that's the end. So I leave you with a picture of my group now. And here you can find more the papers and stuff. And if you have any questions, you are always welcome to write me to this email. Thank you very much.
We have time for several quick questions.
Yes.
So you could also have self-organization of your particles and guide them, say, using evanescent way, and you would get the same result. And you would have actually this well-defined distance between them as well.
You mean if you make a evanescent field with some periodicity?
No, you don't even need to make the periodicity. You have the forces acting on the self-organization because of the forces acting.
I don't follow you. Self-organization, you mean in suspension?
Yeah.
use,
On the surface, you will, if you have evanescent wave,
yes,
and if you have your little particles, whatever they are, as
Yeah.
long as you say, as there are optical forces and scattering forces and absorption,
Yeah.
then eventually, if you arrange, if you have certain size of the evanescent field, you'll see that if you skid along the surface, they will arrange themselves in the field.
this is true, this is correct, and it has been done.
Yeah.
In that case, where I see the potential of this is that I can do different things on the same thing. It's very hard to, with that technique, it's very hard to put, let's say, a gold nanoparticle next to it, a quantum dot, next to it, a magnetic nanoparticle. Because otherwise you will need 3 different surface functionalizations for them, and 3 different surface functionalizations on a substrate, which would require a previous lithographic step. Here, you can imagine, this was done, this is the first experiment, but you can imagine we do the same with a micropipette where we inject gold, tick, 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 tick. Then we clean our system, and now we inject quantum dots, tick, 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 where we want. And then and this is a method that we think will enable us to build circuits of different things. It's not a mass production thing, of course, but it will enable us to test different designs very quickly, I think.
Okay, maybe one more question. We're a little bit behind the schedule. If not, let's reconvene in 10 minutes because we are some